So welcome again, everyone. So we are having the second round of lightning talks. We are starting with Ivanel Bramati from the Bridge Initiative. He comes from the DOOR Institute. So please give an applause to give it the welcome.
Hello everyone, my name is Ivanel Bramati, I'm from DOOR, and also I'm the Bridge Technology Ambassador. So what is Bridge? And Bridge is the Brain Research International Data Governance and Exchange Project, and is funded by the Wellcome Trust, and is an international initiative to structure data governance for sharing the brain data. And it looks at the technical, ethical, legal, and also people with lived experience aspects. And the main goal is to map then the main barriers and also region particularities in data sharing. So why here the bridge in Latin America? Now, Latin America is one of the most diverse regions in the world, and even with our financial and also structural challenges, the scientific outputs has been increased in the last decade, and when data is shared internationally, it must follow each country's rules. So we need the data governance reference to rules the managed through the whole life cycle of the data. For example, the collection, storage, the processing, the creation, the sharing, and also the election. For the technology and structural barriers, here in Latin America, we have limited research and development investment when you compare it to Europe and also United States. So also have a limited infrastructure and also like limit personnel, the trained personnel. We have also a low interoperability across the data standards and formats, and also the anonymization and the identification method is not always standardized, and also we have very different level of personal data protection among countries, and we have little awareness of data sharing routines. So about the people with lived experience involvement, the patient engagement in research is growing worldwide, and the participation in Latin America is still incipient. But there are some initiatives like panels and talks with patient associations and involvement people with lived experience and increase the legitimacy and social relevance of the research. About the legal and also ethical aspects, each country has his own legal framework, and also the ethical review is different between the countries, and also the level of data protection. So this image shows the difference of levels of data protections across the countries. Here also we have vulnerable population like indigenous and traditional groups that need spatial and specific safeguards, and also the defining of sensitive data is still a challenge, and also the data protection levels like very widely in Latin America. So Chile is a pioneer because it had like narrow rights to its own the Constitution. So the opportunities and future plans of the project that many countries are moving to GDPR, the level rules like the General Data Protection Regulation, that's the European Data Protection Law, and also the regional initiatives that can guide the secure data exchange in neuroscience, like to create a regional framework, and this is aligned and is a key to create a common framework to share the brain data in a safe way. So this project, the bridge, support this common data governance reference. So to more a little bit more about the bridge project, you can access our site, and we have here email, Dr. Fernanda Mol, and also this is a survey that you can help in us to map all these questions. So always you can visit our poster, and we will be very enthusiastic about the project, and you can help us to increase this. So thank you.
So thank you, Ivanel. Which poster do you have? Which number? 8. So visit the poster number 8 for more information. Now Nicole Salgado from University of Chile. She's the core facility manager. So please, Nicole.
Hi everyone, I'm going to tell you today about the MicroMundo project, or as we fondly call it, the Google Earth of microscopy. So MicroMundo is a virtual platform, user-friendly, aimed to serve for high school students. And it's a library of microscopy images where are a very high resolution images where students can draw, annotate, and also measure, with the aim that microscopy is more accessible for them without the payment of any hardware infrastructure. Okay? So how did we do this? The use of the platform goes together with school activities. We created a kit with some regions, stainings, and slides, and also a guide of protocols that were given to the schools. And the teachers also were trained in order to perform correctly these activities. And then after that, some samples were processed in a research level laboratory, and the slides were scanned in a slide scanner, and then the whole images were uploaded to a server where they connect to the platform, the MicroMundo platform, where it can be openly accessed. So in the school activities, the students can actually compare their results in the classroom with what they can see in our platform. And the results were pretty nice. Students agree that they can feel more close to science in general. We have some of their comments here, like I like microscopy because I saw plenty of things that cannot be seen otherwise, for example. And in general, it was a very good approximation to microscopy for students and schools that cannot afford that. So this went so well that actually what we are working on now is to expand this project in order to acquire now samples not only from students but also from amateur users, scientific users, and the acquisition of the images can be done in the lab, but also we are working on portable mobile devices to acquire images in order to include them in the MicroMundo library. And this also will be in work with several research institutions. And also we are aiming to focus on samples of environmental, educational, and phytosanitary interest. Therefore, for example, to work with SAG, which is the entity in charge of customs. When you go to Chile, you have to go through customs to prevent the insertion of certain pathogens. So we could work with them in order to create a wider repository of images that can be open accessible, and also with INACH, which performs research on Antarctic organisms. And in the future, the aim is actually start to work with the communities like LABI or other communities in order to make this accessible not only for Chile, but for the rest of the region. We could work in collaboration to access to samples along Latin America. And this platform is fully in Spanish. Therefore, it's a kind of resource that there is nowhere else to be found. Usually, all this is in English, right? So it can be merged with teaching in schools, and also access for research, or a big repository in the end for Latin American specimens and information. So if you, sorry. Just in time. So if you want to see the amazing results of this study and what are we aiming for, please visit us at poster 33, just in front of the entrance of the auditorium. Or you can also, reach us in our social media or at micromundoteam.cl. Thank you very much.
Okay. So the next one is John Ortiz, from PollenGeo, an open software for semi-automated acquisition for images.
Hi. Yeah. Okay. Yeah, it's working, yeah?
Sí.
Sí. Okay, hi everyone. Thank you to Lavi for the travel support. And I want to talk a little bit different from different field, that is geology. And why geology? I'm geologist, and I did my master's degree in geoinformatics, and I'm connected these tools. And now, but we are studying the rocks, let's say in the same computational, we need to use the same computational tools to study the rocks. And here in this case, this is the pollen fossil. The science that I study the pollen fossil is called palynology. And now it's not bioimaging, but by sure some million years ago it was alive. And this is a petrographic study of the rocks. In this case, we took the little skin of the rocks, and then we study under microscope. This is a tomography. We took the rocks and put in the tomography, and this is the kind of image that we are using in geology. The problem that we face is the same, more or less, that the rocks has not cells, has not neurons, but has minerals. and minerals. In this way, we need to do the same problem. We need to identify the minerals, we need to do segmentation, we need to do classification of these minerals, and this is related really close to the field that most of the people is working here. This is a heavy, another kind of mineral. Heavy minerals, we separate from the rock the minerals and need to study the same like morphology of the grains and the volumetric, basically different indices of morphology of the grains. How it is connected with my field? In my master thesis, I developed a software to study stratigraphy. Stratigraphy is the relationship that we study for sedimentary rocks. This is an open source software that was developed in my master thesis. And here we have the representation. It's to do analysis over this kind of sedimentary rocks. In this case, this is already published and was maybe 10 years ago, but now I'm interested in study this kind of image to extract automatically information to help to complement this tool. That is take image data to correlate with the stratigraphy rocks. Okay, this is a brief introduction to the geology imaging, and this is one specific case that we are doing with the Smithsonian Institution, and the Smithsonian has the largest palynological collection from the neotropics, and we want to preserve this collection. We are doing in a digital format. Why is important the pollen? The pollen is important to understand. The vegetation in the past, the paleoecology. We use palynology to date the rocks in different ways. Honey production is really useful. The study of the pollen. Also, it's beautiful. No, these kind of shapes and everything is really, really beautiful. How we digitalize this collection? We took 20 grains. We digitalize in different microscopic techniques these 20 grains, and also in confocal we took 5 grains for each species. This is more or less the data set that we produce. The project has been running for 3 years. This year is almost finished, and the data set will be published, and it's a really well curated and standardized data set. It has more or less 600,000 pictures. It's digitalizing 15,000 species. It's a really large data set. My role in this project was develop one platform that was able to control all the data acquisition process. Is more or less putting one application in the server that is connected with the microscope and is tracking all the progress of the process. The information is stored in a database, a really well curated database, and after that is connected with the microscope, is keeping all the progress during the data acquisition process. Yeah, and here we can track here all the process like user, connected with all the microscope. And something really nice is that the tool is able to help to rename the files, to keep consistency in the names and everything. Here the platform is able to take the name of the default from the microscope, connect with the database, and automatically produce the files in a really nice nice and consistent way. And we have a poster at the end of the right corridor, and my colleague Andres, he has a t-shirt from PollenGeo project. He knows really more details allow the acquisition data process of the data. Okay, thanks.
Okay, so we don't have a number of the poster, but it's at the end of the corridor. Okay, so if you go to the bathroom, you will see it. So I think we go first with Agustin. Mauricio? Sí? Okay. So Mauricio Cerdá is going to present us the SASIVA 2 system from the University of Chile. An applause.
Hi, thank you for the invitation. I'm very happy to be here and present you the SASIVA 2 system. I will explain a bit what is this system about. So first, I come from Santiago, Chile. I think this is one of the most beautiful pictures you can get from Santiago, and you will see it only once per year, more or less, after the strong rain. I work at the Faculty of Medicine. This is the Faculty of Medicine at Universidad de Chile, and my group mainly works in image processing. And one particular thing of my group is that I have a background engineer. So I come from the Faculty of Engineering, and my group is a mix of profiles. So there is a lot of people coming from the engineering faculty, but also people coming from biology, from medical science. So we all the time move between both domains. So what was the motivation for this system, for this SASIVA 2 system, is that as a computer engineer, I'm perfectly fine with this kind of interfaces, with text interfaces, but this is not the case for most non-IT users. See? And on the other hand, if you look at this in the Chilean ecosystem, there are systems, for instance, there are supercomputers available. There is a really nice high-speed network in the country, but there was missing something in the middle, somewhere where you can store your data and do some like initial computations before going to the big clusters. And that's what we propose with this SASIVA 2 system. So let me show very simple two use cases that we have in our system. One is a cloud. So this is open source software called OwnCloud. It's similar to Dropbox, and I don't think I have to explain too much what is that. So you can share files in your group, and it works. And another use case we have in our system is we implemented virtual machines for the users. It means that you can activate a machine with a given characteristics, memory, CPU, GPU, whatever. You can activate it and give full access to their storage by remote. So in which, without terminal, just Windows, whatever the user asks us to use. So, okay, this is how to use it, but what really is SASIVA? So it has been evolving, but at the beginning was something like that, with a bunch of servers in a dark room somewhere in the lab. And we were lucky enough to get funding, so we moved it to a more professional infrastructure some 10 years ago, and now we have this SASIVA 2 system that is the most serious infrastructure here. It's about 600 terabytes of SSD level, that means it's fast, and has GPUs and 5 general use servers. But it's not just hardware, it's a system. It means there is a lot of software going on, and there is a lot of personnel. So we have all what we should have. I mean, we have most of the equipment is coming from Dell computers, and we have all the industry-level software available in this platform, like VMware, NVIDIA, etc. And we set here a bunch of applications that are ready to use for biologists of health domain use. And all this is run mainly by two persons, the sysadmin, who is Malcolm, and myself. So you don't need a super large amount of personnel to run this. So let me talk briefly about the impact. So what are our main advantages? This morning we hear about cluster cloud computing. So mainly I will say we are cheaper because we have money from the government, and we have local support. We have a lot of users, around 400 users from OwnCloud, just to name a few, and our users are very happy. And one of the things they highlighted the most is that they can use this infrastructure from anywhere. So you don't need any specific computers. So you want to know more, please visit poster number 29, and maybe other 2 posters that you will be interested to visit are 19 and 38. That are both solutions that work on SACIVA. Let me, before finish, just thanks our sponsors, the university, the ANID who financed this project, and of course our users. Thank you.
So, thank you, Mauricio. So now we continue with Agustin. Agustin Corbat is a local organizer that will talk about GloBIAS society. So, please.
Thank you. Well, I'm gonna, as briefly mentioned, I'm gonna talk about GloBIAS. It's the Global BioImage Analysts Society. So it's a non-profit association for bioimage analysts. And as I did describe very well, I'm from here. I started 2 months ago. I came back. So GloBIAS aims to pursue worldwide the activities that were previously promoted by GnoBIAS, the network of European bioimage analysts. So it grew a little bit. And it aims to provide all bioimage analysts with a community and a sustainable base through this formal scientific society. Yes, it works. It began a couple years ago, 2023, with a Chan Zuckerberg initiative. That's how it started, and this funding will run out at the end of this year. But don't worry, it's going to continue. So it started. We are now a legal association. So since October 2024, in Austria, this was made formally made a society, a non-for-profit society, and this scientific society aims to connect all this community of professionals that are applying or working with bioimage analysis. To make it sustainable, there's a membership nowadays, but it has regional prices. So depending on the region you're at, you will have different prices. This is more of an estimate. And we already have 60 members, and we had a formal first General Assembly now in June 2025. So this leads to the follow-up slide where I showed a bit of the faces that are here. So this was the executive board that we voted at the mid-year. Well, you can check the president. There's a coordinator, deputy coordinator, and both treasurer and deputy treasurer. Many of this is, there's been a huge effort from Krista Walter. She's the community manager from GloBIAS, and these are both the auditors that are checking that GloBIAS is actually working as it should. And the scientific advisory board is being voted as we speak. I think it's going to be open for a month for every member of GloBIAS to vote who's going to be in the scientific advisory board. Let's go a bit to the goals and the main missions from GloBIAS. So we aim to promote the knowledge and share and advance the bioimage analysis tools, not only the tools, but also the best practices within the field. It goes back to developing and sharing high quality educational material and resources for all skill levels, fostering the networking and professional development, and tackling these big challenges and seizing opportunities in the field. This is a sort of a more detailed list of everything that we've been working on. So it's advancing the quantitative bioimage analysis, promoting collaboration and standards, but there's also developing lists of educational resources, supporting professional developments, seeing what are the emerging challenges in bioimage analysis, and this one is a particular one I like, is establishing a career pathway for bioimage analysts, because as some might know, it's quite an early career and it's not really well stated in many countries, and leveraging this huge amount of interdisciplinary expertise, because bioimage analysts actually come from many different fields. Everybody speaks a different technical language besides the languages from their home countries. So we are organized into work groups, and that's how we tackle the different tasks we have to do. We have a teaching and education work group, communication, the survey 2024, that's it's one of them where I'm leading. Society implementation group, this one it's closed, I think. Workshop 2025, it's about to be closed because it ended at the end of October. And yeah, there's one for publications, conferences, and the HPC and cloud computing for image analysis. Some of the achievements we made is having our own identity, our own website, doing a survey worldwide, although the representation was not really well spread. The workshop and conferences for 2024 and 2025. We are holding seminars, regular online events, in-person events, the YouTube channel with plenty of material, publication of materials, and actually founding this non-for-profit association. So if you want to know more about GloBIAS or discuss about teaching bioimage analysis, you can come to poster number 1. You can check here the QR code or contact at info@globias.org to see everything else. Thank you.
So Agustin is number 1 of the poster, so go and check it. One of the things that Agustin forgot to mention is that GloBIAS gives support for image analysts. I think that that you can register to be helped with your problem with anything in bioimaging. So, and now we introduce Mariano Soiza, Array Tomography Network. So, please. An applause.
Hello. I use this one. Yeah, I think I use this one. Hello, everyone. So first, I would like to thank the organizers for giving me this spot, although it's kind of challenging to be between you and lunch. So I'll try to be very, very short and sweet. Not as sweet as dulce leche, but okay. Let's see. Yeah. Okay. Okay. So let me introduce you. We have I'm part of a network that is called Array Tomography Training and Network. We are funded, basically, we started this project like 3 years ago already, and it's funded by Chan Zuckerberg Initiative, as well as LAVI at the beginning, I would say. And it's, I would say it's a joint effort to try to democratize, in particular, some techniques related to volume electron microscopy. So, it's very hard to say from here. I shouldn't complain because I live in this building, so this is awesome. So the idea here is, in general, you have after you go through ultramicrotomy, you can have volume imaging, and for this, targeting 2D or 3D structures, basically. And then you have, in general, you have 2 flavors. One flavor is collecting these serial sections, ultra-thin sections, in a solid support. That's called array tomography solid support. And then you have another way that is using a tape collector. Okay. In this, we've seen already many studies showing like the fly connectome and this kind of a massive high throughput approaches. So basically, the idea is to be able to combine approaches using light microscopy and scanning electron microscopy, basically combining them through CLEM approaches. So this is the people. Sorry, this is a little low. Okay, so this is the people. We are basically covering most of the globe. We have people from Europe, US, Japan. We are still missing African partner, but basically, and also Oceania. But we are pretty much a nice potpourri of people. So all with the same objective. Okay, this is the distribution. Doesn't matter. Some of us, we are imaging scientists. Some of us are researchers with labs and doing research in brain, in different tissues, but all with the same goal. This is to compare probably the first volume EM analysis that is using the block phase technique that is using FIV and scanning EM, but basically destroying the sample, so then you have a one-way analysis, while in techniques like ray tomography, the sample is preserved and you can come back and reanalyze different stuff, okay, in the same sample. You don't destroy the sample. So then also has offers many opportunities, like you can use different levels of resolution, different levels of actually scaling of your sample, but this is more technical, and also is very helpful because has modularity, meaning that you can go step by step, and at some point you can arrest that point, that step, and you can, for instance, ship samples to another country, and then they can move on with that sample from there, okay? They can catch up on the processing. So then these are a few examples here using from Cristina Micheva, most of them, and from Martina and Thomas in our network, but a human neocortex using light microscopy with array tomography. Then here some images using CLEM, combining fluorescent array tomography with the scanning EM, labeling different synaptic buttons and pre- and post-synaptic partners, okay, in the same ultrastructural volume. And then this is using the atom-tom approach, meaning with the collecting the samples in the tape, okay? So they have typically thousands of serial sections. Okay, so our goals basically was first to set up an online platform that we could put all the resources and share regions and experience with all the users abroad. So then we started in 2023 this arraytomography.org website. Please go and check, because if you want to start with this technique, you have their pipelines, protocols, troubleshooting, a lot of it. And then the second part of it, it was very important. It was the part of training new users of this technique to expand the capacity of this technique. Again, worldwide. So last year we organized a workshop in Munich. It's a hands-on workshop. And this year, starting tomorrow, actually, we are organizing one workshop here. So it's the follow-up of the lobby, okay? We are very happy about it. What else? We started a focus group, a focus interest group, basically discussing sample preparation, serial ultramicrotomy, mapping, acquisition, and a lot of image analysis and segmentation and alignment and this troubleshooting on this. Okay, but then the final goal then is to, as a community, as Lavi probably, I've been learning through all these meetings that the idea is to have a community to share and to grow together, and for that is key to be able to share our expertise in different steps of the technique, also to find a way of exchanging tools and resources and regions. And we are trying to do that, and also to teach and to train people to try to expand our human toolkit, I will say. And then what things we have to put some effort, extra effort perhaps, is in developing tools for image analysis, in developing more selective and precise probes to analyze different components of the biological tissue. Also to play with the substrates that we use to acquire the images, and finally to develop, this is especially together with industry, to develop new equipment more suitable for the work we need to do in the biological tissue. And with that, I think lunch is ready. Thank you.